My name is Jorge A Mayorga, and this is my presentation on my project for microfluidic application testing for genomic research. As Sophia stated, I interned at Standard BioTools, and I worked under the supervision of my mentor, Mackenzie Bullock, and the Research and Development Department. So, formerly known as Fluidigm, Standard BioTools is a biotech company that's based out of South San Francisco, California, that focuses on the design, manufacturing, and sale of biological research equipment involving microfluidic technology. These applications can be used to run NGS library prep or real-time PCR for all types of genomic applications. And although they have several applications available, my main focus in this internship was on the real-time PCR for gene expression and genotyping applications. So since I was mainly focused on running PCR, I got to see firsthand just how many useful applications you can run with it. You're able to run a large amount of assays and samples through a wide range of medical and scientific procedures, which can be very beneficial. However, running PCR all the time can be very expensive, as the applications or the hardware can cost you tens of thousands of dollars, and the reagents that you use can cost you between $500 to $600 for a simple 5 milliliter bottle. So with this in mind, Standard BioTools has created and used advancements in microfluidic technology in order to create their IFC chips, which are used for genomic applications and create a much more time-efficient and cost-efficient workflow for different types of PCR testing. And this can be seen on the table on the right, where I have a comparison between a traditional 384 well plate and one of their IFC chips, in this case, the 96 by 96. So in this example, we are running 1,000 samples on 96 targets, which will equal 96,000 total reactions. And as you can see at the bottom, there is a very drastic and large difference between the number of runs you'll need, the amount of master mix you'll need, and the time to collect that data. So this big difference just shows you how beneficial these IFC chips can be for PCR testing. So Standard BioTools has been heavily investing their time and resources into releasing a new IFC chip variant. And as it goes with anything new, you must do a lot of research, data analysis, and testing in order for this to be a successful launch. So with that in mind, the overall project goal here was to finalize and complete the testing for the 24 by 192 IFC chip, which can be used for all types of genomic applications. This specific chip is intended for customers who would like to run more targets or assays in order to have more thorough results when in comparison to the other IFC chips offered. And for me personally, my personal objective here was to be in charge of carrying out the validation testing for the new application and in assisting in data analysis to make sure that everything is working within the passing parameters. And aside from this, I also had a side project where I was involved in modifying the standard thermal cycling procedure of these PCR runs. And this was in order to find a middle ground as to how fast you can accelerate and speed up the cycle speed procedure. So just as a precursor, I had spent the first 2 to 3 weeks of the internship familiarizing myself with the procedure for preparing these IFC chips for gene expression. And I did this by running several practice runs prior to the official testing. So since this is a validation,
I think if we're not going to go with data,
since this is a validation study, the way this runs is it will consist of me running the same performance test 9 times independently with a 100% success rate in order for the entire study to be considered a pass. So how I started was I first made my assay mix in a 1.5 milliliter tube with all of the reagents that follow. And after that, I made my sample mix, which will be 2 separate samples, will be your 1x sample and your 2x sample. And essentially, they're the same sample in the fact that they have the same DNA and the same amount of overall volume. But the difference here is that the 2x sample has double the DNA concentration, which will be necessary for your results later. After this, I prepared the IFC chip by using loading reagents, which included the control line fluid, the pressure fluid, and the actuation fluid. So once we have this ready, we then added the sample mix and the assay mix into the chip, and I included a loading map on the left, which shows you where to add each liquid and each reagent. Once the chip is ready to go, I then loaded it onto the real-time PCR system, which you can see on the right, which will run the gene expression procedure for us. After about 2 hours, the procedure would be over, and then I would analyze my data using the Standard BioTools PCR analysis system. So before I explain the results, let's first go over the passing specifications and just kind of what constitutes a passing test. So as you can see on the left, we have 4 parameters that each test must independently pass in order for this to be a success. So for the first specification, we have a delta CT, CT standing for cycle threshold. And essentially what the delta CT is it's the difference in the sample abundance between the sample of the 1x sample and the 2x sample. And this must be between 0.9 and 1.1. After this, we have the standard deviation, which is the variance in the uniformity of the sample abundance, and this must be under 0.2. After that, we have the no call rate, which basically just means it's the amount of reaction wells within the IFC chip that didn't react or produce a result. And lastly, we have the average runtime, which just shows us that it must be under 2 hours and 15 minutes. So now that the parameters are out of the way, let's go ahead and look at the results. And as we can see, we had a 100% success rate, which is exactly what we wanted. All the data and all the numbers were within or under the specific specifications, which is a great thing. So in conclusion, we were able to complete all 9 tests successfully within the set parameters. The software, the hardware, and the chemistry were all able to work together with no issues, which is exactly what we wanted so that the customers can have an easy and reliable procedure, no matter what assays or samples they use. So therefore, the 24 by 192 IFC chip will be ready for release with the gene expression application. And as far as the next steps go, from here, we will write and finalize the official report, and we're going to hold a formal data review with members outside of the research and development team so that they can double check our work and verify that everything is correct and it works as we say it does. And lastly, the product management group will then prepare for product launch in October of 2023. So as far as key skills gained, I learned a lot from this internship. I learned how to write and analyze industry standard lab report. I developed a greater understanding of genomics and gene-related testing. I gained genuine lab experience, and I learned crucial research and development skills. I gained a greater knowledge on microfluidics and the biotech industry as a whole. And some major challenges that I faced was meeting project deadlines and handling multiple projects at a time. Getting my initial understanding of microfluidics and reagents, I'm not going to lie, was pretty hard for me at first, but I got the hang of it after a while. And we also had an issue where we had an intensity issue on one of the PCR systems where it was just, it was reading the dyes incorrectly. And we were able to solve that as well with the help of other departments. And as far as personal insights, learnings, and takeaways, this internship gave me the confidence to pursue undergraduate research at UC Santa Cruz as I applied for the course-based undergraduate research there. It allowed me to not be scared to ask for help or ask lots of questions. And it also just gave me the confidence to do research independently. And thank you to everyone. Thank you to SLI. Thank you to Marissa and Sophia. Thank you to my mentor, Mackenzie and Michael, for helping me out. Thank you to Standard BioTools for giving me a hand. Thank you to Chrisman Yañez, all my friends and family, and Dr. Ron Painter, who is the professor who recommended me.